All right, so think about what we have done. And remember, as we go through this course, I told you that we continue to build on everything that we've already talked about. So think about all of the testing for our patients that we've talked about so far. We've talked about your patient ABO, all right, investigating any ABO discrepancies. We talked about performing the IAT to see if there are any unexpected antibodies in their plasma. And when I say unexpected, that's other than the ABO antibodies, okay? Those are expected, all right? When we say unexpected antibodies, we think about other allo antibodies such as any of your Rh antibodies, Duffy A antibodies, Duffy B, JKA, JKB, okay? Antibodies to other red cell antigens, those are all unexpected. All right, and we talked about performing a DAT to see if there had been an IgG antibody or complement attached to patient cells occurring in vivo, right? That's occurring inside the patient's body. We are now moving on, we're adding on another test that is performed on every patient that comes through blood bank, and that is the Rh determination. All right, now I do have a disclaimer there. There's some language. So if you say, what Rh type are you? That really has a connotation of referring to the D antigen, okay? So, but the Rh also terms the whole family. That is a whole family of antigens, all right? So it has the D antigen, but also big C, big E, and little e. That is your Rh family. There's some other rare ones that we'll talk about a little bit. But those are the main ones. That is your Rh family. So while the D has a connotation of being referred to as the Rh, and you're either positive or negative, all right? Some of you guys might have heard, oh, I'm B positive. That's what that positive refers to. Do you have the D antigen on your red cells? If the D antigen is there, you're Rh positive. If the D antigen is negative, then you're Rh negative, okay? So that's what lecture 8 is going to talk about. We're going to talk about the Rh family system, D, and then the other antigens in that family as well. But I just wanted you guys to understand, some people use the term Rh just to describe the D antigen, so don't be surprised if you hear blood bankers describe it like that. All right, so the Rh blood system, like I mentioned, involves, the whole family involves multiple antigens, and it's a very complex system, and it's very highly polymorphic. And meaning polymorphic, meaning that number one, the structure is very polymorphic, and there's also different genes involved, right? So it's not just the D antigen, it's also the C, the little c, the big E, and the little e, all right? So that means it's highly polymorphic. It is the second most important red cell antigen after your ABO antigens, okay? The D antigen is very antigenic. I cannot stress to you, this is why we have RhoGAM, all right, for Rh negative females that become pregnant. She will get RhoGAM to prevent the formation of anti-D. It's kind of like a prophylactic treatment. That is the only antibody that we have prophylactic treatment for. And as we talk about KEL, if you guys, KEL is the next antigenic after D. And so if any of you guys can invent a prophylactic to prevent pregnant females from developing KEL, then you will be very rich, all right? But it hasn't been developed yet, all right? So only D, and the reason we have RhoGAM is because D is very antigenic. It is very highly capable of causing hemolytic disease of the newborn and transfusion reactions. So if you lack the D antigen, remember Landsteiner's rule, if you lack the D antigen, you can build the antibody to it, all right? So if you are D negative and you're exposed to D positive cells, whether that's through a transfusion or for females, if your fetus is D positive, then you can build anti-D. And it will, just a very small amount can cause hemolytic disease of the newborn for pregnant females, and also for those recipients that have received the transfusion, okay? So as we move on in this course, keep that in mind too. The main concern about that, so you think, well, what's the worst case adverse reaction that that patient's gonna have, is they're gonna build anti-D, right? For females, huge concern for their future pregnancies, okay? We don't want that anti-D to attack the baby. For males, it's not so much of a concern, all right? So in trauma situations, we hoard our O negatives, okay? O negatives are like very priceless, very precious in blood bank. So if we have a male that has been involved in a motor vehicle accident and he's type Rh negative, we might actually give him Rh positive units, all right? Because he is a male, the only thing that's going to happen is he's going to build anti-D, okay? And the thing is that anti-D is an IgG antibody, all right? So it's not going to happen immediately, all right? He is going to build that antibody. The problem will occur for future transfusions, all right? Does that make sense to everybody? That would be a amnestic response, okay? There's actually more than 50 related antigens that have been included in the Rh blood system. Again, we're only going to concentrate on the 5 major and most common ones, and those are D, which is we do in our routine testing on every patient. Okay, that's your ABO and RH. All right, that RH type that we do in our forward testing, that's your D. All right. Also in that RH family is your big C, your big E, your little c, and your little e. Okay, those are the 5 most common antigens in your RH family. Now, when this is called phenotyping, when you are identifying the antigen on your red cells, that's phenotyping. All right. If you figure out that there's an antibody in your patient plasma, then you would phenotype your cells for your patient cells for these. All right, because one of the things is your patient has to be antigen negative in order to build the antibody. So as you start identifying these antibodies, if you think your patient has anti-big E, we want to phenotype our patient to make sure that they are lacking the E antigen on their red cells. All right, so that's kind of, so just remember D is done in routine testing. Every patient will be tested for the D. These are only done if you suspect the patient has the corresponding antibody, okay? Right, so your RH family was first recognized in 1939, and the RH name was given to the system because the antibody was made by rabbits and guinea pigs, and it was found to react with the rhesus monkey. So that's where the RH terminology comes from. It's named after the rhesus monkey. And then really, it's the RH antibody, all right, so we're really thinking about the D antibody here, reacted with about 85% of human cells, all right? So think about that. The RH antibody reacted with 85% of human cells. That means 85% of the human population have the D antigen. You guys need to know that, all right? 85% of the population is going to be D positive. 15% of the population is going to be D negative, all right? Right, and like I mentioned, the D antigen of the RH antigen, D is the most important. It is very antigenic. All right, so here's a little comparison of your ABO antibodies that we've talked about previously and the RH antibodies that we're moving into. Remember, ABO antibodies are of the IgM classification. They can react at room temperature. They are naturally occurring, all right? So remember, the ABO antibodies after about 6 months of age will develop from the food you eat, the air you breathe, the exposure to bacteria, all right? They're naturally occurring. Your RH antibodies are all IgG. They only form from exposure, like from a previous pregnancy in females or from a transfusion, other donor cells, okay, that have the antigen that you lack. Okay. ABO antibodies can react at room temperature. You're going to see that our RH antibodies, they do not react at room temperature. They will only react at 37 degrees and AHG testing. Okay. All right, there's some different theories on the inheritance of RH antigen. The current theory explains genetic control of the RH antigen expression has been enhanced with the ability to characterize the amino acid sequence, which is controlled by the genes, all right? So we talked about that in the genetics of immunohematology, that the genes control the DNA is in your nucleus, messenger RNA will copy that DNA pattern, and then transfer RNA brings in the amino acid, which produces the protein, which then controls the expression of the antigen on the red cell, okay? right, so the most widely accepted theory describes two close-linked genes, so at the same loci on chromosome number 1. All right, so you have one loci that controls the D antigen, all right, and then you have the other loci that controls the C and the E expression. Okay. All right, so RHD is the terminology used to express your D antigen. D negative people lack the D antigen, and in blood bank we have a terminology that we use for that. So your patients can be D positive, meaning that they have the D antigen. So you're either D positive or D negative. All right, if you're D negative, blood bank has a terminology that we use. It's designated by the little d. There is no little d antigen. All right, this is an amorph, kind of like group O, right? All right, so you're D positive, meaning that the D antigen is being expressed, or you're D negative, and blood bank notates that with a little d. All right, again, I want you guys to understand this is an amorph. There is no little d antigen. That's why on your IAT screening cells and your panel cells, all you see is D, okay? You don't have D and then another column with little d. You're not gonna see that because there is no little d antigen, right? That's just the way we notate it, meaning that your patient is Rh negative. Okay? So if you're D negative, then you have no genetic material at that RHD loci, okay? RHCE determines your big C, little c, big E, little e antigen expression. And so here, if you think about what that means is, here's your two different loci. Notice here's your RHD, and this is on chromosome 1 on the P arm. And then here's your RHCE, and notice that you have two. All right, so on each chromosome you have two genes. All right, so if you have the D expression on both of these, then you're going to be Rh positive, right? If you have the D expression on one and not the other, you're still going to be Rh positive. You still have the addition of D antigen on your red cells. Okay, if you lack the D antigen at both of these sites, that is when you are Rh negative, okay? And then here's your RHCE, and so you could have big C and big E being produced. You could have big C, little e, you could have little c and big E, and you could have little c and e. Does that make sense to everybody? So on one allele, you could be getting big C and big E. On the other allele, you could be getting little c, little e. That means if you have this two combination of alleles, you're going to be heterozygous. You're going to be big C positive, little c positive, big E positive, little e positive. Those antigens are going to be expressed on your red cell membrane. Does that make sense to everybody? Versus if you had the allele combination Rh big C big E, and then you have Rh big C little e. Notice there's no little c being expressed in that combination, so you would be little c negative. All right, does that make sense to everybody? Okay. And then you can use this to determine your frequency of progeny. So if you know the dad is the D antigen is only expressed on one allele, then he is what we call big D, little d, he's heterozygous. All right, one allele has the D antigen being expressed, the other one does not. And the same for mom. So then 25% chance that the baby will be, well, actually, this patient would be Rh positive, okay? This patient, even though only one allele is expressing the D antigen, still Rh positive. Okay, and then this is your Rh negative genotype. All right, so you have to lack the D antigen, both alleles to be Rh negative. So 75% chance this patient's offspring will be Rh positive. Okay.
So if you don't have the D antigen, can you not express CE?
You can. So they're inherited independent of each other. So you could be D negative, but you could be big C, big E positive. So they're inherited. There are two different loci, so they're inherited separate. They're not dependent on each other. Does that make sense? Mm-hmm. But they're inherited like together. This, like if you inherit, this site is on one loci. So this whole chromosome will be inherited, one from mom, one from dad. All right, so the D is independent, but the C and E travel together.
Is there any, like with the CE groups here, is there anything like resembling dominance, or it's just as if you've got the alleles, it's going to express? Like,
that's right.
Okay.
Yes.
Is there any?
Because you really have to think about, there's really no little d antigen.
Right.
This just notates that one of the alleles is not expressing the d antigen. And that's how we notate it with the little d. Aaron, did you have a question?
Is there any like adverse effects for having little e and little c, or big E and big C?
So if you have the antigens, you're actually fortunate, because in the event for female pregnancies, or in the event you're transfused, if you have the antigen, you're not going to build the antibody. The problem occurs if you lack these antigens and you're exposed to them, and then you build antibodies. And as we start thinking about frequencies, most people are more likely to be little c and little e negative. And so if you build those antibodies, it's more challenging to find compatible antigen negative blood for you. So if you have the antigens, you're good. You don't have any worries. If you lack the antigens, then you could potentially build these antibodies. That's the only adverse reaction side effect from being, you're better off being antigen positive than antigen negative. I don't know if I've told you guys this, but there is a guy, his name is Dr. Joe Chaffin. He is a pathologist. He is known as BloodBank Guy. If any of you guys have followed him, if you're on social media, follow him. He's a pathologist. He's a huge advocate for medical laboratory scientists. He actually trains and educates pathology residents in transfusion medicine. So he has a lot of podcasts, a lot of YouTube videos, and he talks about all topics relating to blood bank. So if you guys ever need further information, he's a great resource. And again, it's at BloodBank Guy. He's on Facebook, he's on Instagram. He has a website with all types of informational videos. I actually had the pleasure of meeting him at a national conference. I heard him speak. So that was total blood bank nerd moment. So if you guys want to follow him, you can. But I took this picture from him. It kind of just demonstrates possible inheritance patterns. All right, so notice that your RHD, which controls the presence of your D antigen, and your RHCE, all right, it's inherited. This is a whole set, all right? And so you'll get one from one parent and one from the other parent, okay? And so this just kind of further lets you know. Notice that this offspring is big C positive, little c positive, big E positive, and little e positive. So that this patient's actually Rh positive for all of the antigens. And then he is, the D gene is being expressed on both alleles, so he's Rh positive. Notice that here, this patient, we haven't talked about the Wiener language yet, but little r designates that your patient lacks the D antigen. All right, so that's a different terminology that we're about to get into. And notice that the D antigen is not being expressed there. Okay. All right, so you have big C and little c, they're codominant. Big E and little e are codominant alleles, and then D is the dominant because there is no little d antigen. Again, it's an amorph. All right, so here's your gene frequency. We already mentioned that 85% of the population will be D positive, 15% of the population will be D negative. You guys need to know that I would not be so much concerned memorizing the other ones. I would like to point out that 98% of your population is going to be little e positive, right? Remember what I talked about, you're better off being little e positive because if you are little e negative, you're in the 2% of the population. Can you imagine trying to find compatible antigen negative units in 2% of the population? Like our donor pool to find E negative units is very small. All right, so that's why, and this is a real world situation. It happened to me. If you are in the blood bank on the weekend by yourself and you have a female come into the OR and they are asking for blood and she has anti-little e, that is a nightmare for a blood banker by herself. All right, so I'm screening every unit on my shelf. I have a Red Cross reference lab. I have called them in order units, and meanwhile the OR is screaming at me. All right, so 98% of the population is E positive, very hard to find compatible blood. All right, so in blood bank, you guys have been to the blood bank, you know that we kind of hoard. When we find units that are antigen negative, we hoard them for as long as we can. All right, so if we find a unit that's little e negative, we're going to put it on our shelf and save it for our patients that we know have anti-E, or in the event of a trauma, we can pull that unit.
You're making this blood bank internship sound
You guys,
incredibly more appealing.
it's fun though. Like it was very stressful, but it's like problem solving, time management. It puts everything together. It's just so, and I say I was by myself. I had a lab assistant, all right, who manages the front desk. And you have the ability to call in. Somebody's on call. So if you have a mass casualty or, you know, earthquake or something and all these traumas are coming in, of course you're going to call in people. But that's a real world situation. It happened to me, all right? So the higher, the more population has the antigen, the harder it is to find antigen negative blood, all right? Okay, so there's some terminology, you guys, and most students hate this, but you have to be able to convert one terminology to the other, all right? So there's actually two systems that we're gonna talk about. There's actually two additional systems that it was developed more for entry into computer systems, all right? It's all numbers, and so I'll mention those, but your Fisher race and your Wiener nomenclature, you have to know how to transfer those, okay? And it's all based on inheritance patterns. All right, so here's the four terminologies. Fisher race uses the DCE terminology, all right? Your Wiener nomenclature uses the RHHR terminology. So we're gonna use big R, little r, numbers 1 and 2, and then prime and double prime, okay? We'll get to that in just a minute. Rosenfield, again, was a numeric terminology, and then the International Society of Blood Transfusion decided that there were too many red cell antigens, and so they started giving all of them numbers, all right? And so you guys don't have to worry about memorizing all those numbers. Your textbook references them a little bit, but it's really how some computer systems program them in for each antigen. All right, so this just breaks down the current inheritance theories. Here's our current, where the D allele is at one loci and the C and E are at the other, okay? And then here's your possible alleles. Here's your Fisher race, says that D has its own loci, big C, little c has its own loci, and then big E or little e is at another loci. So this actually has 3 loci on each chromosome. And then the Wiener terminology states that the D, C, and E is all expressed on one loci on the allele, okay? Okay, so your Fisher race states that there are 3 loci involved. Each gene produces an antigen, okay? So you have your D, you have the big C or the little c at one loci, you have the big E or little e at that loci. And then here is how the terminology is written. So if one chromosome expresses let's say, take this example. The D antigen is being expressed on one allele. The little c antigen is being expressed on one allele, and the big E antigen on one allele. Okay, so this is one allele. The slash separates the two alleles. All right, so then your second allele would be D negative. All right, notice that you could see it like this. All right, or you might see the little d included there. Right, and that just indicates that this allele lacks the D antigen being expressed. Big C positive, little e positive. Okay, so this is your Fisher-Race terminology. You might also see it written in alphabetical order. All right, so some people might see, you might put C, D, and E. All right, so you might see it as that. I find that to be confusing. I personally like it like this. Okay, so but just know sometimes you might see it written like that.
But on the test, you'll have it written as the first way.
Typically, yes. For this course, I try to use I'll use it this way. But just be aware, you might see it written like that. I mean, it's the same thing, like little c, big E, and then the D antigen being expressed. It's just written in alphabetical order. But I think this is used more widely, so it's just more familiar to me, maybe. All right, and then here's your Wiener terminology, stating that it is one loci that will determine your alleles and then the antigen specificity, okay? And then here's your terminology. You actually have 8 different possibilities. Big R means that the D antigen is present, and this is read RO, okay? So if you have RO, that means that only the D antigen is being expressed. So then you'll have little c, little e, okay? R1, actually, I will explain this in a future slide, okay? We'll come back to this. I think it'll be easier. But these are your possible combinations. It uses big R for D. Notice little r means that the allele lacks the D antigen, and then these subgroups represent your little c, little e, big E, little e representation. Okay. So what you have to be able to do is, if given a Fisher-Race terminology, the DCE terminology, you need to be able to convert it to your Wiener terminology. Okay. All right. So I want to give you guys a really good way to do this. If you guys have that chart that was on that slide, you see the whiteboard, Sarah? Okay. I want you guys to think about this in positions, all right? So your Fisher-Race has 3 positions per allele. This position will be either big D or little d, okay? Fisher-Race. Which will be notated in Wiener language as big R or little r, okay? This location will be notated as, in Wiener location will be 1 or 2, okay? C or little c, E or little e, big E or little e. Okay. Think about this as position 1. This is position 2. If the big antigen is present in position 1, so this would be if you're big C positive, then, and let's say, let's do an example of DCE, all right? The D antigen is being expressed, the big C antigen is being expressed, the big E is, well, that's, oh, let me do this. Let's make that a little c. okay? Think about this as position 1, and this is position 2. All right, so this is D. So what do we notate that for in our Wiener location? Big R or little r? Big R. Now, if big C was present, then it would be R1. Okay, because big E is present, it's R2. Okay, so it's notated like this. Does that make sense to everybody? Okay, what if it was big R or little r? Big R. 1 or 2? And this is 1. Does everybody see that? Okay. What if you had little d, big C, little e? Big R or little r? Little r, 1 or 2? 1. Oh, actually, okay, this is confusing. When you use the little r, we don't use 1 or 2. We use what we call prime. All right? That's a prime. That's kind of like an apostrophe. What about D, little c, big E? Big R or little r? Little r. What do you guys think? 2 primes. So that's red, little r, double prime. Okay? All right, so don't worry, we're going to get practice with this. We have plenty of examples we're about to go through. And then, if you have, let's say you have D, little c, little e, what do you guys think about that? Big R, little r? What do you think about that? Yes, R-O. And then sometimes if you have little d, little c, little e, sometimes that's just written as little r by itself. Okay. What if you had big D, big C, big E? That is RZ. That means your position 1 and position 2 are both being expressed. Right? Big C and big E is RZ. Right? What if you had little d, big C, big E? That's little RY. Don't ask me where the Z and the Y come from. I'm not exactly sure why they chose that notation. But that's what they did. Fisher or Wiener did. Does that make sense to everybody? And again, we'll get plenty of practice.
Why did Wiener make up a new system?
Wiener came up first. He was in 1943, and then Fisher-Race came behind him in 1948. And it all has to do based on their inheritance theories. All right. So Wiener thought that, and you guys can't see my screen. Wiener thought that it all came from one loci on the allele. So
why don't we all just...
Now, why he chose big R, little r, where that notation came from, I do not know. Fisher-Race makes a lot more sense, if you ask me, and it's a lot more easier for students to understand the Fisher-Race terminology. But you have to be able to convert it to the Wiener. I promise you guys will probably see a question on your board of certification about that. And if you ever work in BloodBank, then you will need to be able to understand. I don't know if you guys noticed it or not, but on your panel cells, they are categorized. They're all group O, but it will give you your Wiener notation. So like if you have a, what we call an R2 cell, they have the highest D expression, and we're going to talk about that. It will let you know which cells are R2. And I can show you what that looks like on the IAT. Sarah has a question. Sarah asked if we need to worry about CW. So CW is actually included here. You guys see noted here. It is what we call a private antigen. If you guys look on a panel, most of your population is going to be CW negative. And it's just not very common for people to build an antibody to it. So it's what we call high titer, low avidity. I doubt you'll ever see it. So CW is not as of concern. Big C, little c, you'll see that a lot. All right. And really, CW is really not included in your Wiener. It's not included in your Fisher race or Wiener terminology. So I don't have to worry about it in that aspect. Okay, so let's do some practice. And I printed these guys out for you. I want you guys to try to figure these out. So let's do the first one. Big D, big C, little e. All right, so what do you guys think about big D being expressed? So we're going from Fisher race to the Wiener terminology. All right, so big D, big R. All right, and what about the big C being expressed? R1. Okay. And then what about this guy, little, the absence of the D, antigen, little c, little e. Just little r, all right? So this is written as R1R, okay? Sometimes you might see it written without the slash, okay? It might just be R1, little r. What about the next one? Big D, big C, little e. That's still R1. Okay, because your big C is present. And then what about this guy? Big D, it's the same one. All right, so that's R1, R1. What about the next one? Notice that the little d is absent on both alleles. All right, so we just have little c and little e. What do you guys think? Little r, all right? Okay, this one, what's big D, big C, little e? R1. What's big D, little c, big e? R2. All right, so that's R1, R2. What about big D, little c, big e? R2. What about little d, little c, little e? R. So that's R2, little r. Here's one. What is if the D antigen is absent, but we have big C and little e? R, little r prime, okay? And then what's little d, little c, little e? Little r. And then what if D, big C, and big E are being expressed? That's your RZ. And then the same is being expressed on the other chromosome, the other gene as well, so that's RZ as well. Notice that that patient is negative. This patient would be negative for little c and little e because both alleles are expressing big C and big E. All right, so this patient's big C positive, big E positive, little c negative, little e negative. Okay.
Is not having the little d there the same thing as not having it there at all?
Right. Yeah. So you're written like this?
Versus the DEC.
Right. So it could be written like this, where the little d is included, or they could totally leave it off. Yes. And then the last one, we have the D antigen absent from both alleles. Then we have C and E, big C, big E being expressed. Little RY. Yes. All right, does that make sense to everybody? Right, I want you guys to spend some time on this. You'll have some study questions for more practice, but you have to be able to translate these terminologies, okay? Your Rosenfield terminology uses numbers. So like the D antigen would be a 1, the C antigen would be 2, the big E antigen would be 3, little c is 4, and little e is 5. And then a plus or minus sign is included to determine if those antigens are present. So it looks like this. Or I guess it's just a minus sign. All right, so if you have a patient that is D positive, C positive, E negative, little c positive, little e positive, that would be written as if D is your 1, right? So you have 1 there. Big C is notated as 2, so you have 2. Notice that your patient is big E negative, so you have minus 3, all right? And then little c would be 4, and then little e would be 5. This, not used so much, all right? More Fisher-Race and Wiener terminology, more common. Here's another example. You have the D antigen, little c, big E, so the presence of 1. Notice that big C is not being expressed, so it's minus 2, right? Big C is your 2. 3 is your big E, all right? So it's positive. 4 is your little c, it's positive. And then 5 is your little e. Okay. So that's another way to write it. Does everybody see how you can figure out what your patient is positive for? So take a look in this example. This patient D positive or D negative? D positive. Is this patient big C positive or big C negative? Big C negative, because neither of these alleles are expressing the C gene. All right, so the C antigen is going to be absent from your patient red cells. All right, what about big E? Is this patient positive or negative? Positive. What about little e? Your patient is little e negative. Okay. So that's another type of question you might be given Fisher-Race language and say, is this patient little e positive or little e negative? Okay, and you would say little e negative because neither one of these alleles are expressing the little e antigen. Okay. Remember, genotype is the actual genetic makeup of the material. Phenotype is the outward expression. And so you can use this for genetics testing to determine your offspring probability. So here's your frequency of the most common haplotypes as they are inherited in both the white population and the black population. So notice that for the white population, having the D antigen, so being Rh positive, with big C and little e in Wiener location or notation, that's the R1. So that's the most common haplotype present in the white population, right? Most people, because 85% of the population is going to be Rh positive, they're going to have the D antigen, they're going to be big C positive, and remember 98% of the population is little e positive, all right? So they're going to have the little e antigen as well. Okay. So this comes into play if you're ever trying to let's see, when would you use this? Let's think about when we do our D-testing in our serological techniques, we either know if they're Rh positive or negative, right? We don't know if one allele is lacking the D antigen, right? In our testing, they're still going to be Rh positive. So in our serological testing, we can't really determine for the D allele being expressed on both antigens or only on one allele, all right? So you can try to take the race of your patient and figure out what their most likely phenotype or genotype is. Okay, so this is kind of where that comes into play. There's a disclaimer on that. I said that we can't really tell. You're either, Rh positive or negative. There are some, what we're going to talk about, weak D expressions, and there's some variant D expressions that we'll talk about. All right, but for the most part, you can use this to kind of figure out what our genotype is of our patients. All right, so how do we know what the antigen expression of our patients is? Well, we do phenotyping, all right? And so anti-D is included in our ABO and RH determination. Like I mentioned, it's done on every patient. And we have commercial antisera, which is your anti-D that you guys will actually get to do today in lab. All right, we're adding on the RH. And then you have antisera that is manufactured to detect specifically the epitope of that C antigen. All right, and so big C, anti-little c, anti-big E, and anti-little e. All right, so that's how we determine if our patient is expressing the antigen on their cells. And so here's an example of where you can kind of try to figure out the genotype of your patient. So if you have a patient that's typed as D positive, big C positive, E negative, little c positive, little e positive, then your phenotype, we know the D antigen is being expressed. It's heterozygous for C, so it's big C positive, little c positive. Notice that it's big E negative, so it lacks the big E antigen, and little e positive. So there's a couple different combinations. If you look at the white population, big D with big C and little e is the most common occurrence. So it's probably that one. And then also your patient is little c positive. So the next most common would be this. So that's kind of how you can use your frequencies to determine the genotype of your patient, based on the antigenic expression. For the black population, using that same antigen typing results, notice, okay, your patient's heterozygous for big C, and he's Rh positive. We don't know, just because he's D positive, we don't know if both alleles are expressing the D antigen, okay? So he could actually be, oops, big D, little c, little e, or he could be D, big C, and little e. Could be that combination. Why is he most not likely this combination, this one with this one? Because he needs the big C, right? And so he's probably this one, all right? All right, so we have some patients that we classify as weak D. And what that means is there's a couple different reasons that can cause a patient to be weak D. And when we say a patient is weak D, that just means that the D antigen expression on their red cells is very much lowered, that sometimes our reagents can't detect it. So when we start talking about performance of our D testing, we have what we call the weak D test. And so your first step in your D determination, just like your front typing and back typing of your ABO, initial spin, read for agglutination. If it is negative at initial spin, we want to determine if our patient's what we call weak D. So if you get a negative reaction at initial spin, you will take your weak D tubes. You're going to put them in the incubator, incubate for 37 degrees for 15 minutes, read for agglutination. If it is still negative, because remember, anti-D is an IgG antibody, given our monoclonal determination of our antisera, we can detect the anti-D. However, sometimes if it's a very weak expression, we miss it. So enhancing that antigen antibody complex at 37 degrees can sometimes help us see that agglutination. Right? Synthesization might have occurred, but we're unable to see it. All right. So then after 37 incubation, we will spin and read for agglutination. If it's still negative, we are going to add the AHG reagent, okay? And we're going to perform our AHG test. At the time you add AHG, that is when the test becomes weak D, okay? So if you have a AHG positive reaction after being negative at initial spin in 37, then we call our patient weak D positive. He's still typed as RH positive, all right? You might see it written as DU. Most commonly, that's an old terminology, DU. Most everybody will now use weak D terminology, right? They're going to call it weak D. All right, so what are some causes of this weak D phenomenon? Number one, it could be genetic. Maybe you inherit a weakened expression of the D antigen. A really good an example of this is one allele has the D antigen, the other one does not. So what that means is if you think about your patient red cells, the D antigen is directly on the red cells. If you're having both alleles express the D antigen, there's going to be all these D antigen sites on your red cells, right? Our antisera has plenty of binding sites to bind to, we'll get agglutination. If you're heterozygous or one allele lacks the D antigen, right, you have fewer D antigen sites. Not as many antigen binding sites for our antisera to bind to. Okay, so that's a weaker D antigen expression. Okay.
Do we have like statistics on that? Like how many people who are, let's say, heterozygous for D?
I do not know the statistics off the top of my head. With the advancement of monoclonal antisera available to identify the D antigen, and remember the D antigen is very complex. It has different variants, and so there's different epitopes. And our antisera has become more likely to pick up on all of those epitopes as well. So we're not seeing as many weak Ds as we used to as advancements have been made in blood bank reagents. Aaron, I'll try to look up that statistic for you. All right, so there's another reason why your D might be weakened as well, and it has to do with the location on the alleles. All right, so if you're in the translocation, if the D antigen is inherited on the opposite chromosome of big C, that is known to weaken the expression of your D. And it's just thought that the big C being on the opposite chromosome weakens the D expression, okay? Now, in the cis location, when D is inherited with big C, D is very strong, all right? So if your patient has big D inherited in the translocation from big C, this patient might be what we call weak D, all right? So here's an example of what that looks like. Right, so if you have one allele, you have D antigen expressed with little c, okay, and then big C is on the opposite allele, that would cause a weak D reaction. And in this example too, D and C are here, but on this allele, it's opposite. All right, that will cause a weak reaction. Versus notice that in this situation, in the cis position, D and big C are together, the other one lacks the big C. All right, so this would be a regular strong RH reaction. All right, so I took this from your textbook. It just kind of breaks down in the Wiener nomenclature the strength of our Ds. And your D deletion, that's what this is notated as here. It's either notated as D dash dash or dash D dash. And what that means is only the D antigen is being expressed. Your patient is negative for all the other RH antigens. Big C negative, little c negative, big E negative, little e negative. All right, so that's your D deletion. Because the D antigen is the only one being expressed, you have very strong D reactivity with that one. All right. And then R2, what is our R2 terminology? What does that break down to? So it has no presence of the big C antigen being expressed, right? And so remember, when C is inherited in the translocation from D, that weakens the D, all right? So the D is actually very strong in an R2 cell versus an R1 cell, okay? That's because big C is absent here, right? So that makes the D stronger. Okay. I showed you guys this because this comes into play when we start talking about titering and choosing our cells to determine a titer for anti-D on a pregnant female. You will always want to choose the cell that has the strongest D expression. So we will always choose the R2 cell, all right? This will come into play a little bit later on. So there's a third reason why you might have a weak D expression, and it is what we call D mosaic, or what we call partial D. And that is where, remember I mentioned that your D antigen structure is very complex. What happens is, in this scenario, your patient might be missing part of that D molecule. And so what happens is our antisera cannot bind to it, therefore we are unable to detect it. I mean, there's a lot of molecular-based research going on right now, and I think you guys are going to do a molecular activity. You guys, have you already done the ABO determination? You guys, are you doing a RH determination too on your patients? You guys do that? But you could use that same principle, what you guys did in BloodBank, to determine your patient's RH. There are tons of D variants being identified on a daily basis right now. It's huge molecular research going on right now. And this is why, because that D antigen is so polymorphic. Some people lack the whole antigen. And then so what you'll see with this guy is he can test as D positive, but he's also going to have anti-D in his plasma. All right, so anytime you see that, you should automatically suspect some type of mosaic D, partial D. All right. If you suspect that, you might want to use a different manufacturer's monoclonal anti-D. Remember, monoclonal because it is specific for a specific epitope. All right, so if you use a different manufacturer, you might be able to get different reactions. So how do you type your patient if you determine that your patient is weak D positive? Remember, the weak D test begins at the addition of AHG in your testing. So if you get a positive reaction with AHG, that means your patient is weak D. They are still RH positive. So if you're doing patient testing, you type your patient as RH positive. If you're doing donor testing, very important that all donors have the weak D test. If they're a negative at initial spin, they are typed as RH positive. Okay. It is only if you get a negative reaction at AHG do you type your patient as RH negative. All right. So they're typed as RH negative. They can be typed as RH positive. If they're weak D positive, they can be transfused with RH positive units. Okay. And then for moms, they do not get ROGAM. Right, because they just have a weak D expression, but they have it, right? So they will not get ROGAM. There's a phenomenon, it's called RH null, and that is when the patient lacks all of the RH antigens. So they're D negative, they're big C negative, they're little c negative, they're big E negative, and they're little e negative. The problem occurs with that is if you think about your red cell membrane, and I took this from your textbook, the RH antigen is thought to play a huge role in your structure of the red cell membrane. So if the RH antigen is completely missing, it's thought that there's increased phospholipids, causes the cells to shrink, and it can cause the formation of stomatocytes on the peripheral smear. So this would be very similar to hereditary stomatocytosis. All right, you're going to see a lot of stomatocytes. They're going to have an increased osmotic fragility. It's going to result in a hemolytic anemia, very similar to hereditary spherocytosis. Okay, and so it's thought the phenomenon of Rh null causes the shape change in the red cell membrane, and that will result in hemolytic anemia in these patients.
So can you have Bombay and Rh null?
I have never seen a documented case of that, but not to say that it can't happen. So this would cause a hemolytic anemia in your patient. So you're going to see all of those symptoms: decreased haptoglobin, increased retics, increased bilirubin, LD. Right, so you'll see all the symptoms of hemolytic anemia. All right, so let's talk about our week D testing. What exactly do we do? There's 3 different types of reagents that have been used. All right, so you have your slide or your rapid tube. We have what we call chemically modified IgG antisera, and then we have what we commonly use today. We have our monoclonal polyclonal. All right, so here's your slide in rapid tube. I don't know if any of you guys have ever seen this before. You have like this plate, you add your reagents, you add your patient sample, and then you'll read for agglutination. Most places aren't doing this anymore. This will be called your slide test. And most places aren't doing this anymore. Okay, when you perform this test, we will always perform I don't want to say always. Sometimes you will use a Rh control. And the reason for that used to be because of the use of high protein reagents. Remember that high protein can cause false positives. All right, so usually we would add a control in there just to make sure that our reagents were not causing the positive reactions. All right, and so you guys in the 9B lab, we will always use an Rh control. And what that means is the Rh control contains everything that is in your anti-D reagent except the antibody. All right, so it has all of your diluent, everything is the same except it lacks the antibody. Okay, so when that comes to play a very important role, think about if you have an AB positive patient. What's your front type going to look like? Positive with everything. Anti-A is going to be positive, anti-B is going to be positive, A comma B is going to be positive, D is going to be positive. How do we know that that's not due to some type of polyagglutination, something attached to the patient cells causing those positive reactions in all of those tubes? The reverse is negative, but that's using patient plasma. Yes, but you want to make sure that our reagents are not causing all of those reactions. So we can use what we call a Rh control. That gives us a negative reaction in the front type, letting us know that our patient's reactions are true and not due to polyagglutination. If your Rh control is positive, that invalidates your test. All right, we have to figure out why that Rh control is positive. It might involve washing the patient cells and then repeating the test. If those cells, maybe there's some excess protein present in the patient cell suspension. All right, so we could wash and repeat. We could try to use different antisera. Those are some examples. And then we'll talk about the difference in just a minute. And we also have chemically modified IgG antisera, IgG antibody. That means it's clinically modified at the hinge region. And that just means that it opens it up. It allows for better binding. This is associated with fewer false positives, again, because you don't have that high protein reagent that you're using. But you still want to use the Rh control for the AB pos patients. All right, and then what we commonly use today, what you guys will probably see in every lab that you go in, you have your monoclonal IgM and your polyclonal IgG testing. So it's a blend. The monoclonal, polyclonal, and the addition of the monoclonal IgM is why we are able to detect the anti-D at room temperature, initial span, okay? The polyclonal combination IgG lets us detect it at the AHG phase of testing. All right. And again, this is the most commonly, this is the best reagent to use because of that combination. But you still want to include that RH control if your patient is AB positive. All right. All right, so week D testing, what it looks like. You have your D testing is going to start out at initial spin, just like your normal ABO testing. All right, you're going to add anti-D to your patient cells. You're going to spin and incubate, or spin and read for agglutination. If that is negative, you will take your anti-D tube. You're actually going to add another drop of anti-D, because what happens if you forget to add anti-D? You get a negative reaction, right? So you could have a false negative because you forgot to add the reagent. So we're just going to add an additional drop of anti-D. And remember that zone of equivalence, serum to cell ratio, we're adding a little bit more antibody to further encourage the antigen antibody complex, right? To make sure that our antibody will bind if that D antigen is present on the red cells, okay? Then we're going to incubate. It's very important when I say add an additional drop of anti-D, you add it to your D tube only, not your control tube. What happens if you add anti-D to the control tube? It could turn out positive, all right? So we want our control tube to stay negative, all right? All right, so after incubation, you're gonna spin for agglutination and read it. If it's still negative, then you're going to wash both tubes. Do not set up new tubes. You're using the same tubes throughout this procedure. You're going to wash 4 times, either by hand or using a cell washer, and then you're going to add AHG. Spin and read for agglutination. Remember, when you add AHG, you're going to read macroscopically for agglutination with your eye. And if it's negative, you're going to confirm it under the microscope. We're going to look for microscopically positive agglutination. All right. If you get a positive reaction at AHG in your patient D tube, then you call your patient weak D positive, RH positive. All right. If you get a negative reaction in your AHG tube, then your patient is called RH negative. Okay. What must we do to those 2 tubes that have AHG in them if they're negative? We must add check cells. All right, and remember what check cells are. They're IgG coded cells. You're telling me that you have a negative reaction, so all of that AHG in your test tube is unbound. The check cells will bind to that AHG. It confirms a negative AHG reaction. All right, that is not reflective of your patient testing. That is just an internal quality control. Okay. If your control is positive at any time throughout that test, your test is invalid. All right, and we're going to look at some scenarios of when that would happen. If your control is positive at the AHG, what that means is, think about what we're using. We're using patient cells combined with AHG. What test does that sound like? Not the IAT, that is your DAT. So if you're adding AHG and you get a reaction, what does that mean? Positive DAT, something is coding patient cells, all right? Therefore, that positive DAT is interfering with the ability for us to determine an appropriate RH on our patient. We call that an invalid RH. Okay, we have to do further investigation. Number 1, if you have a positive control at AHG when doing the week D testing, probably going to do a DAT, going to be positive, then you might have to do an elution. There is a treatment, and I think I have it on a future slide. Oh, I'll talk to that in just a minute. All right, so here's some scenarios. If you're doing your testing and your RH tube is positive, your control is negative, what do you report your patient as? RH positive. What if your RH tube is negative and then your control is negative? What do you report your patient as? RH negative. What if your RH tube is positive after AHG phase of testing and your control is negative? How do you interpret that test? Your patient's weak D positive, they're RH positive. Okay. What if after AHG phase of testing your RH tube is positive and your control tube is positive, that is an invalid RH. You can't report out that patient's RH, okay? If there's a trauma situation and you have an invalid RH and you're currently working on that sample, the OR is screaming for blood, you can always give negative. You can give RH negative blood to these patients until you get that RH figured out. All right, if you have an invalid weak D test, there's a treatment that we can do in blood bank. It's called the EDTA glycine test. And what it does, it's very similar to an elution, except for, if you guys remember when I talked about an elution, I said the red cells were no longer viable because most elution processes destroy the red cells. All right, they lyse the red cells in some matter, whether they're using acid or heat treatment. All right, this does not lyse the cells. It dissociates using a very low pH. It dissociates the antibody from the antigen. All right, so whatever's causing that RH control to be positive in our weak D testing, we can pull off that antibody. And now we have viable red cells that we can get a valid RH type on. Okay, does that make sense? So you'll take patient cells, you'll do your EDTA glycine treatment, you can use those red cells, you'll repeat your RH testing. Well, number one, you'll want to do a DAT to make sure that DAT is negative. That negative DAT means that their antibody is no longer coating the cells. All right. Then you could repeat your weak D testing and get a true D type on your patient. Right, does that make sense to everybody? The only caveat to that is EDTA glycerin is known to denature the KEL antigens. So it destroys the KEL antigens on the patient's cells. So if you're doing any type of antigen typing, you won't be able to get an appropriate KEL representation, right? Because that antigen site has been destroyed.
Would you be using this sample for KEL testing if you're already trying to confirm a weak D test?
It depends, because if you have a positive DAT, that means something is coating patient cells. You could have a positive DAT and have a negative IAT. So, and then anytime you do have a positive DAT, you have to do an elution and figure out what's coating patient cells. Potentially, you could, depending on what's coating patient cells. If the IAT was positive, you could. But what you could do is, depending on the antisera that you're using to type KEL, you'll have to read in their process to see if it uses AHG phase of testing. If it uses AHG phase of testing, you don't want to use patient cells because it could cause a false positive, right? Because something's already coated in those cells. In that case, best scenario is to give KEL negative units, just to be sure. Okay, so like anything, we're concerned with false positives and false negatives. False positives for your D testing. If that patient has an autoantibody, all right, that will cause your RH control to be positive. Okay, so we'll pick up that. Rouleau, okay, will always cause false positives in our testing. Contamination, if the reagent has been contaminated with other reagents. Polyagglutinative cells, all right, if there's something coating your patient cells, extra protein or anything like that. And then failure to follow directions, okay? False negative, the number one reason false negatives, forgot to add your antisera, all right? So that's why we always add our antisera first. Want to make sure that we don't have false negatives due to lack of adding the antibody. Neutralization of reagents, all right? So we've talked about that before. If your AHG, if you have all that excess protein, improper washing can neutralize your AHG, will cause a false negative. If you use the wrong antisera, right? If you add anti-D to your control tube, that could cause a false positive. Okay, if you add control to your RH test tube, that could cause a false negative as well. All right, so make sure you add the appropriate reagent to the tube. If your cell suspension is too heavy, that would be too much antigen. All right, and then if you shake your button off too hard, of course. All right, so let's talk about the characteristics of our Rh antibodies. Remember, our Rh antibodies are classification IgG. They result from prior exposure, whether that be females from a previous pregnancy or exposure to a previous transfusion. Sometimes you might have patients that had both. They had a previous pregnancy and they had a transfusion in the past. So in that case, we don't know where that antibody might have came from. This is why patient history is so important. Blood bank will always look at patient history. Our Rh antibodies are typically of the IgG class, and they usually do not involve complement. However, they may show dosage. So an example of dosage, if you had a heterozygous cell, big C, little c, and it reacted weaker with your patient plasma, because it's sharing the antigen sites, big C and little c, that will give you a weaker reaction with your patient plasma. Versus a homozygous big C, if the antibody is big C, will give us a stronger reaction. That is an example of dosage. All right, so there's some rare antigen expressions. CW, it's included in your big C and little c. Only about 2% of the white population and almost no blacks have this. Like I mentioned, if you look at the panel, pretty much everybody is going to be CW negative. So to have the antibody is very rare. Little f is what we call a compound antigen. It's found on the cell when both little c and little e are present. And we had a patient here at UAMS that had little f. So it was rare. It looked like he had anti-little c and anti-little e. So the way you treat these patients is you would give them little c and little e negative blood. And you guys know 98% of the population is little e positive. All right, so to find little c and little e negative blood, very challenging. Okay, you'll probably have to call in specific donors. And then you have G. It's located on the red cells that possess either the D or the big C antigens. So if your patient is negative for D and big C, they're also negative for big G. So if they're negative for it, then they can build the antibody to it. So in this situation, we would want to give them D negative, big C negative. There's another one, V or CES, found in 30% of American blacks, less than 1% of whites. Again, so these are really rare. Again, I've only seen the F one time. But I want you guys to know about them. All right, so that is the Rh system. It is very complex. I think you guys see that. So spend some time on the Fisher race and the Wiener nomenclature. Make sure you can go back and forth on those. We talked about the weak D expression. You guys will actually get to do the weak D test this afternoon, so it'll make more sense to you after you actually do it. And then make sure that you're aware that the Rh testing sometimes will include the Rh control. You guys will always do it in this course. Don't be surprised if you go to the clin lab and they're not including the Rh control. However, if you have an AB pos patient, you will always have to have that Rh control.
So if the control is positive, the Rh control is positive, would you do an elution?
Probably so, because if your Rh control is positive, you're going to do your DAT, starting out with polyspecific AHG. If it's positive, then you're going to do monospecific IgG, monospecific complement to see exactly what's coding patient cells. Anytime you have a positive DAT, you have to do an elution. So.